Hello, everyone. My name is Dr. Jennifer Ziegenfuss, and I'm a postdoc here at UMass Chan Medical School. And I am happy to introduce you all to our topic for today, which is the microbiome, which is a fun topic and part of research that is becoming even more interesting with more and more things being discovered about how it's involved with our overall health and diseases. So this is the Science Cafe for UMPA, which stands for the UMass Postdoctoral Association. And I just wanted to let everyone know straight off the bat that the views in this presentation and on all the slides that you'll see today are solely those of the presenter and not necessarily those of UMass Medical School. So continue with that. Today we're going to have two talks by Dr. Benedikt Mortzfeld and Sage Foley, who is a PhD student here at UMass. First, Dr. Mortzfeld, he is a postdoctoral associate in the lab of Dr. Bucci in the Department of Microbiology and Physiological Systems. Before joining Dr. Bucci's lab at UMass, he obtained his PhD at Kiel University in Germany. Benedikt is interested in how bacteria, antimicrobial peptides affect ecological dynamics in the gut environment and how that knowledge can be used to engineer probiotic beneficial bacteria that can target relevant human pathogens. So his talk today is entitled Repurposing Weapons of Bacterial Competition to Promote Gut Health. And Sage's talk will be Heroes of the Gut Microbiome that help maintain a healthy intestine. Sage Foley is a seventh-year PhD student in Beth McCormick's lab here at UMass. Her work involves investigating the gut microbiome and which components are essential or detrimental to our health. After graduating with her doctorate, Sage wants to continue her research in the growing microbiome research area in the biotech sector. So, before we start our talks, I just want to remind everyone to please type your questions in the chat box rather than asking questions during the talk or after the talk. So please, at any time, just put your questions in the chat box and then we'll get to them at the end of the talk. And before we start with Benedikt, I want everyone to mark their calendars for the next Science Cafe seminar, which is Wednesday, October 27th at 6:30. Okay, and then I'll have Benedikt screen share.
Yeah, thank you very much. Can you see my slides? Perfect.
Yeah, looks good.
So thank you very much for the kind introduction. I'm a postdoctoral associate in Bucci's lab at the UMass Chan Medical School. And today I will be telling you about repurposing weapons of bacterial competition to promote gut health. And I want to start off with telling you that bacteria are really everywhere. So you can find them in fresh water, ocean water, in the soil, on plants. You can even find them in very hostile environments like Chernobyl, where basically everything else is getting killed because of the radiation, or at the bottom of the sea in hydrothermal vents, where bacteria are basically living in boiling water. But it's not very surprising that bacteria can do all of these things because they have been around for almost 4 billion years. So up here. And all the eukaryotic organisms, which are the ancestors basically of all the plants, animals, and humans that we know today, they are at least 2 billion years younger, which means when the eukaryotes arrived or emerged, they had to cope with the bacteria that were already around, and they had to find ways of living with them or even cooperating with them. So I want to ask you, what do you think, how many bacteria do we live with approximately on our body? So our body consists of more individual cells than we have bacteria in our microbiome. Is that a true or a false statement?
All right, 10 more seconds for the poll.
Great. Yeah, 2/3 of you are correct. So this is a false statement. So it is estimated that we have at least as many bacterial cells living on us. How do I? Yeah, at least as many bacterial cells living on us as we have human cells. Probably it's much more. So the bacteria are covering all our surfaces that are connected to the environment, which includes our mouth, the airways, the gut, and in total, it's approximately up to 100 trillion bacteria in total. And most of our bacteria, most of our bacteria, of our symbiotic bacteria, we receive from our mothers during birth or during breastfeeding. And approximately by the age of 3, our large intestine is one of the most densely populated areas on the planet. And our bacteria in total, all our microbiome can weigh up to 2 kilograms, which is 5 pounds. And they provide us with very important functions, including interaction with our immune system, breaking down some of our food, providing us with essential compounds, or even protecting us from pathogens or epithelial injury. But it's very important to remember that the symbiosis between us and the bacteria can be very different. So in general, there's a mutualistic relationship, which means we have bacteria in our gut, which I already said, that are breaking down some of these food components and make them accessible to us. At the same time, they can live in our gut and they're pretty happy there and pretty well protected. So it's a win-win situation. But then we have commensal bacteria that are pretty much just living with us and feeding of us, but we don't really have an advantage. You can probably compare this to a barnacle living on a whale. So the whale doesn't really care too much about the barnacle, but the barnacle is getting to different places and can explore different food sources. And then we have parasitic relationships with bacteria, where we usually relate to as pathogenic or infectious bacteria, which means when they grow, they harm us, or they even try to kill us. But even here, it's very important to remember that this is dependent on the context. So if we, for example, have a bacterium that is infecting our lung and is very infectious in the lung, once it maybe reaches a different body site like the skin or the gut, it may not be as infectious as in the lung. So it's very much dependent on the context. And one of the probably most impactful discoveries of the 20th century were antibiotics, which was good because in general, if you cut your finger and you get an infection and you don't want to risk a bloodstream infection, you can treat it basically with antibiotics. And before the treatment of antibiotics, this could be your healthy gut microbiome, for example. The problem is once you take the antibiotics, it doesn't only reach your finger, but it gets distributed all over your body through your blood system, which means it also gets everywhere else. So at the end, your finger is still good, but it may shift your whole microbial community to a completely different state, which may cause secondary problems we're learning more and more about. So basically, think of this as a nice day on the beach. A lot of people are there, everybody's nice and spread out. And it's pretty hard for new people to come in because all the spots are already taken. So this is something that we refer to as colonization resistance. But if you take antibiotics, it wipes clean some of these areas and makes it pretty simple for new people to come in or for some of these pathogenic bacteria to take up the space and grow and cause some problems in your gut. Sometimes they even bring antibiotic resistance genes, which makes it even worse because they are not really affected by the antibiotic that you're taking, but they also start sharing these genes or the antibiotic resistances among this microbial community, which makes it even harder to get back to the initial state. So what would be obviously very nice would be to target these pathogens specifically instead of broadly, to remove the pathogens and not affect the rest of the microbiome. And the approach that we're taking are the bacteriocins that we want to use as selective weapons. So bacteriocins are small antimicrobial peptides that have evolved for bacteria-bacteria competition, which means bacteria are always looking for nutrients, they're always looking for spaces, and they try to kill off close relatives just because they're taking up the same space. And Bacteriocins, these antimicrobial peptides, they act locally, usually right where they are produced. And they can be very broadly effective, which means they're targeting a very central mechanism in the bacterium, which is usually a cell membrane or the cell envelope. But they can also be very selective by getting into the cell and then inhibiting some of these vital cell functions. And into the cell they get with some Trojan horse mechanisms, which allows them to get access to the cell and then bind to their targets and inhibit them. So this works, for example, like this. So as I already said, bacteria are constantly searching for nutrients, like this blue pathogenic bacteria down here. And one of the nutrients that bacteria are always looking for in the gut is iron, because iron is very limited in the gut. So they produce these molecules that can bind iron into the surrounding. And once these molecules bind some iron, the bacteria take it back up and use the iron for the metabolism. But obviously the blue one is not the only one that can do this and that can take up the iron. There's a lot of other bacteria, but this green one, for example, has learned to not just produce these molecules that are binding the iron, but they attach a small toxin to the same molecule, which means the green one is protected because it has an immunity mechanism while it is producing the toxins. But once our blue pathogen here tries to take up some of the iron from the environment because it thinks, because it wants to grow, then it encounters one of these toxins and it gets killed. But this killing mechanism is so specific that it's not affecting all the members of the community, but only specific members. And in this case, it's very selective for E. coli species, Salmonella species, Klebsiella species, and Shigella species. It's important to say that this effectivity is independent of these common antibiotic resistances, because it works completely different and it's a completely different mechanism, which means we can also target some of these multi-drug-resistant pathogens that are making problems. So how this usually works in the lab, when we're testing these bacteria for activity, we have one of our green bacteria here, grow them in a colony, and while they're growing, we let them produce our compounds, and then we add this drug-resistant bacteria to it, but they can only grow in the area basically where we don't have the toxin. So what we get is this area of inhibition around the colony in the middle. And this is exactly what it looks like on our plates. So we have the green bacteria here in the center. They are producing our bacteriocins, producing it into the surrounding. And when we add our, in this case, multi-drug-resistant Klebsiella on top of it, the Klebsiella can only grow where there's no toxin. And so we're currently working towards using this system in actually living organisms, and we're using mice for this. So this means once we encounter the colonization with some of these multi-drug-resistant bacteria in the gut, we would like to add some of our living therapeutics that are basically passing through the gut. And while they're passing through it, they're producing our bacteriocins, so the very selective compounds, killing these multi-drug-resistant pathogens along the way. And since this is very specific, it would not affect members of the healthy microbial community. Yeah, coming to my take-home messages, what I wanted to show you today is that bacteria are basically everywhere, which is not necessarily a bad thing, because most of our relationships with bacteria are either good or neutral. Sometimes we encounter a bad one, which we can treat with antibiotics, but antibiotics are not always the best choice, since they also disrupt our healthy microbiome and can cause other problems down the line. So we're using this approach of bacteriocins as an alternative for antibiotics to selectively eradicate pathogenic bacteria from the gut. And finally, I would like to thank my lab, including my PI, Bucci, who is very supportive of my work and my ideas, and of course our funding sources. And I'm happy to take questions.
Thank you, Benedikt. That was such an interesting talk, and it's such a novel approach to actually treating drug-resistant bacteria. That's amazing. Everyone, please feel free to keep putting in your questions. And while they come in, I have a small question of my own. So what I wanted to understand was, you said these toxins that are produced by the Trojan horse bacteria, they use iron as sort of a carrying molecule. So would there be any healthy bacteria also which would be affected by taking in these toxic iron molecules?
Yeah, so that is a very specific question because we always try to split things into good and bad bacteria. And when I'm saying that we are trying to target like a Klebsiella species or a Salmonella species, not all of these are bad necessarily. I mean, there are some good ones and some bad ones. And also this bacteria strain that we're using to produce these toxins is also an E. coli strain. So it is also very close to, so it is E. coli, it is very close to some of these bacteria, but it's not causing any problems in the gut. So it's, yes, along the way, we will probably also produce some collateral damage of killing off some of these bacteria that are closely related to our pathogens, but it's not at all in the scope of the antibiotics, killing basically whole clades of bacteria.
Okay, Benedikt, I have a next question for you. So do all kinds of bacteria produce bacteriocins, or just a pathogenic bacteria have the capacity to produce the bacteriocin?
No, so bacteriocins are a very general mechanism of two bacteria fighting for space and for resources. So pretty much, so I wouldn't say now like 100% of all bacteria are producing bacteriocins. There may be one or two exceptions probably somewhere, but it's a very general mechanism of bacteria competing for resources and trying to kill each other off. So yeah, so bacteriocin is basically an umbrella term for antimicrobial peptides produced by bacteria target at other bacteria.
Okay, thank you.
Interesting. So Manish has a question. He's asking, do viruses, fungi, and other microbes also live as symbiotically with humans? And another one is, can you engineer some of the resident microbes to produce these Klebsiella killing toxins to eliminate them from the gut?
Yeah, so the first question is a very good one. So yes, in general, the answer is yes. So also viruses and fungi seem to have a symbiotic relationship with humans, but the research is not as far as for bacteria so far, because they are not for fungi so much, but for viruses, it's much, much harder to work with. And also viruses have different targets. So theoretically, viruses can target human cells, but viruses can also target bacterial cells. So theoretically, there are ways of some bacteria producing viruses against other bacteria, and thereby modulating basically different growth patterns. So it's basically adding another layer of complexity. And some people are trying to target it, but they are not that much into it as what we already know about bacteria interactions. The second question was, so if we can also produce or take some naturally or like some native bacteria basically and engineer them. Yeah. So that is basically what we're doing. So we're using an E. coli strain that has been originally isolated from humans, but the one we're using is a transient strain. So what we don't want is something that engrafts long-term and then it's hard to remove again. So we would like to, or we're using something that is basically just transitioning through, and on the way it's killing the pathogens, but after some time it's also diluted out and we cannot find it anymore. Instead of having something that sticks around forever, and then you would have to figure out how to get rid of that one again.
So well, we have a couple of more questions. One from Roxana Pfeifferkorn. She asks you, Ben, is it expected that resistance mechanisms will come up against the Trojan horse toxins?
Yes. So that is always a question that we get, obviously, because we're trying to find alternatives for antibiotics. And the question is, do the mechanisms that we're using also at one point will encounter resistances? And the question or the answer is probably yes. So bacteria basically can do anything. If you just give them enough time, they can probably also develop some resistances. in this case, so what is not the case with most antibiotics is when bacteria can degrade these compounds. The mechanism that we're using is basically specific for the iron uptake, which means that it probably comes with a very big drawback if you develop a resistance mechanism, because you also cannot take the iron from the environment anymore. So you may become resistant, but you also may not be able to grow as well anymore. So it's probably a very big drawback for the bacteria who become resistant to these ones. But in general, certainly, yes, bacteria can develop resistances to anything if you just give them enough time.
Okay, next question we have from Matthew. He's saying, wonderful talk, Ben. I have two questions. Are those bacteria producing toxins found in the environment as well?
So first, thank you. So what does he mean by environment? So environment is always a term. So environment like outside of humans.
Yes.
So far, the ones that we're using, no. So they seem to be very specific for bacteria species that are living in the gut, and the only ones that are known are from gut representatives. But obviously, there's also a strong bias because of all the medical research that is going on, that a lot of the bacteria that you find in the databases are gut isolates or mouth isolates, so human isolates instead of soil or environmental isolates. So there's obviously also a big bias towards that one if you look into the databases. But the ones that we know so far are only coming from human isolates.
Okay, I think that's his second question was actually related to the first one. So if the answer is no to the first questions, I guess it dictates. Thank you.
All right, everyone. So the next talk that we're going to have is with Sage Foley. So Sage, if you would like to start your screen share, that would be great. And Sage is going to talk again about the gut microbiome and what things that we normally have in our bodies might be good or bad.
And can you see the slides okay?
Yes.
Okay. Well, hello, my name is Sage. I'm a graduate student working towards my PhD in Dr. Beth McCormick's lab at UMass Chan Medical School. And my project is studying how bacteria in the microbiome of a healthy gut prevent inflammation. So some bacteria of the healthy gut, as Benedikt introduced, play a role in supporting intestinal health. And this could be by digesting components of food that you consume and turning it into vitamins and nutrients for the human tissue. And sorry, I meant to get my pointer going. Can you see that? Okay.
Yes.
Thank you. So the bacteria in the intestine can receive signals from the intestinal tissue. The bacteria can also communicate with each other, and the bacteria can release molecules to communicate back to the intestine. And this symbiotic relationship resembles that of a balanced ecosystem living in harmony. However, when the ecosystem is perturbed, there could be a shift in the types or amount of bacteria in the intestine, and this shift is called dysbiosis. So first, let's start with a poll. And we would like to see what you think can impact your gut microbiome in daily life. Is it medication, diet, exercise, genetics, or all of the above? Strong response. Like you said, it's 30 seconds, so a few more seconds. Awesome. So I don't know if you can see the results, but most people said all of the above, which is correct. So the correct answer is all of the above, all the things we mentioned and more. So there are many factors that influence the microbiota that we are still trying to understand. But some of these do include human genetics, birth mode and feeding, for example, whether you have a natural birth versus C-section, and then breastfeeding versus formula feeding, the composition of your diet, drugs and medications, including antibiotics that Benedikt introduced, hygiene habits, and exercise habits. So interestingly, over the past 60 years or so, the incidence or number of immune disorders, including autoimmune diseases like type 1 diabetes or allergic diseases or atopic diseases like asthma, has been increasing. And the hygiene hypothesis proposed by Dr. David Strachan in London states that this increase coincides and may possibly be linked to the increasingly clean living habits in parts of the world like the United States. We also just want to mention that during times of a pandemic or flu season, it's still important to wash your hands, but overall it's thought that we're becoming too clean. So in addition to cleanliness, this increase may also be linked to increased antibiotic use, more sedentary lifestyles, and increasing diets that are high in fat. And because these factors influence the microbiome, it's thought that the microbiome may be involved in these diseases. So what we are beginning to understand is that many of these disorders are linked to a perturbed microbiota or dysbiosis. And many of these links are still just correlation, but more and more studies are trying to unravel whether a dysbiotic microbiota actually causes these diseases. My project in the McCormick lab looks at ulcerative colitis, which you may have heard of as a form of inflammatory bowel disease. It is characterized by debilitating inflammation of the large intestine, also known as the colon. The treatments that are currently available only reduce the inflammation, but don't treat the underlying disease. So we are working to understand why disease occurs so that we can learn how to treat or prevent disease. And the microbiota is altered in ulcerative colitis or UC patients. So we are studying whether the microbiota plays a role. So I am studying in my project regulation of a specific protein called P-glycoprotein that is present at high levels in a healthy intestine, but is at very low levels in inflamed tissue. And this protein is important because it prevents inflammation. So inside the intestine, there is a layer of cells that form a nice barrier between the inside of the tube, called the lumen, and the underlying tissue. And P-glycoprotein is present at the surface of these cells, and its role is to pump out molecules from the cells. And underneath this layer of cells is the bloodstream that contains immune cells, such as neutrophils. And neutrophils normally stay in the blood, but can travel across the cells to the lumen, or the inside of that intestinal tube, when needed to fight off infection. But in the absence of an infection, P-glycoprotein releases molecules that block this neutrophil movement to prevent unnecessary inflammation. So therefore, I am studying to understand what keeps P-glycoprotein here at high levels to maintain health. So we hypothesize or believe that it may be changes in the microbiota that impact P-glycoprotein levels. The loss of P-glycoprotein could be due to either a loss of hero beneficial bacteria or a rise in villain or harmful bacteria. And because of the complex ecosystem, we use mouse models to study these interactions. So some experiments that I work on include altering the microbiota with selective antibiotics to reduce subsets of bacteria and see if this impacts P-glycoprotein. I can also reconstitute mice with a microbiota, which means adding bacteria back into mice that lack a microbiota and see if this affects P-glycoprotein expression. So from experiments like this, I've collected fecal samples from the mice which contain these bacteria that we can then sequence to understand which bacteria are present. So from the feces, we can purify out DNA and put this through a sequencing instrument that will determine the order of the bases in the DNA. So different bacteria have different sequences of DNA, that act like barcodes that we can use to infer which bacteria and how much of each are in the sample. So shown here is an example of real data from one of my antibiotic treatment experiments in mice. So each type of bacteria is represented by a different color, and we can see, depending on the antibiotic used, the collection of bacteria are shifted in different ways. Then we can also look at the intestinal tissue of these mice to look for the amount of P-glycoprotein. So in this example, we saw a decrease in P-glycoprotein expression or amount in these mice that were also missing the green category of bacteria due to the antibiotic treatment. So we could deduce that these green bacteria are important for P-glycoprotein to stay present. What we concluded is that a loss of a group of hero beneficial bacteria led to a drop in the amount of P-glycoprotein. So next we wanted to look at what molecules these bacteria could be releasing into the intestine to communicate back to the intestinal tissue. And we found that when we added some of these molecules to intestinal cells in a dish in the lab, or directly to mice, that this showed an increase in P-glycoprotein. So to be sure that what we're finding in the lab is relevant, we can confirm our findings in human patient samples. So we have been collecting for a few years samples from healthy and ulcerative colitis patients during colonoscopy procedures. So we have collected biopsies, which are little punch extracts of tissue that we can look at for the amount of P-glycoprotein. We've collected stool or feces that we can sequence to see which bacteria are present. And we've collected brushings or scrapings of the intestinal surface to measure the amount of that molecule that's released by P-glycoprotein that prevents inflammation. So something interesting that we have found is that when we put feces from healthy individuals onto intestinal cells in the lab, we see that this increases the amount of P-glycoprotein in these cells, which likely means there's something in the contents, either bacteria or the molecules that they produce, that is increasing P-glycoprotein. We don't see the same thing with feces from UC patients. So one next step will be to transfer these feces, including the bacteria and molecules that they produce, directly into mice and see if this changes P-glycoprotein in their intestine and whether this prevents inflammation. So to summarize, we have found that a subset of bacteria act as heroes and release molecules that increase P-glycoprotein in intestinal tissue to prevent unnecessary inflammation. And we are relatively far from a clinically approved treatment, but what these findings suggest are that we can shift the microbiota to increase P-glycoprotein to limit inflammation in UC patients. So future treatments for UC could include delivery of bacteria, and this could be either one bacteria species or a combination of species, delivery of the bacterial molecules that are produced, or a third area of active investigation is how changes in the diet can impact the microbiome. So to end, this is our lab, Beth McCormick's lab at UMass Chan Medical School. People have come and go over the years, but this is a collection of everybody that I've known. And we should get an updated picture soon. And of course, our funding sources at the bottom. I'm happy to take any questions.
Great. Thank you so much, Sage, for that awesome talk. So I'll start with the question I have. So you said the P-glycoprotein can be influenced by the ratio of the good and the bad bacteria as such, but can a bacteria also secrete some molecules and influence the P-glycoprotein level? Is that also a possibility?
You mean can the bad bacteria secrete? So I think the good bacteria in the context of P-glycoprotein, there are some bacteria that can cause an increase in P-glycoprotein and some that can cause a decrease. And both of those scenarios can be through release of different molecules.
Okay.
And for example, salmonella infection, which you may know you can get from foodborne illness that causes a lot of stomach and intestinal upset, that actively reduces the amount of P-glycoprotein in the intestine.
Okay, thank you so much. We have a next question from Manish. And Manish is saying, great talk, Sage. With antibiotic too, you also see a loss of the green bacteria, even though P-glycoprotein was high. What would that suggest? Was there gut inflammation seen in these mice?
Was there inflammation in, an antibiotic too? We didn't see a difference, so I guess what this would suggest is that it might not be a dose dependence of the amount of molecule or the amount of bacteria, but just whether or not they're present.
Okay, thanks. Thank you. So if anybody has any questions, please put in the chat box.
Sage, I wanted to ask something. So, very interesting talk, and it's amazing to know how much you can learn from different samples taken from the body. I was wondering whether what would be the role of the neutrophils that usually move into the gut under normal circumstances when it's not inflammation?
So normally they're driven across by a separate signal that senses an infection. So they shouldn't normally be at a very high level in the intestine, but they will come across naturally if you are infected with something that isn't supposed to be there, to kind of sequester it and get rid of the infection, and then they're supposed to go back into the bloodstream but not stick around. So when you have inflammation in the absence of infection, that could be due to neutrophils coming across when they're not supposed to, and it could also be due to them not leaving when they're supposed to.
Thank you. You also mentioned that there could be certain molecules other than the bacteria that might help in increasing P-glycoprotein levels. What would sort of be the idea with that? Is it just something that increases the bacteria that increases P-glycoprotein, or?
Yeah, so it could deliver either the molecules that the bacteria produce just without having to deliver the bacteria, or potentially deliver molecules or via the diet, giving the right food for the bacteria to then produce the right molecules that are needed for P-glycoprotein. And this could also influence the amount and types of bacteria that are able to grow.
Thank you. Yeah, please feel free to ask any questions in the chat box and we can ask them for you.
Okay, I can ask one question. So to what extent do you think the antibiotics can affect, the P-glycoprotein expression? Is that also could be because of the antibiotics?
That is always a possibility. The specific ones that we tried in mice, we also tried in the lab with just the intestinal cells in a dish, and the antibiotic drugs themselves did not change P-glycoprotein amount. So we think it's the downstream effect of the antibiotic, which is the change to the bacteria.
Okay, thanks.
Interesting, thank you. Benedikt has a question. He's asking, do you know what kind of molecules may be triggering the response? And would it work to synthesize those and give them as a medication instead of giving the bacteria?
Yeah, so we have found at least 2 types of molecules that contribute to this effect. So both are driven by consumption of a meal. One is a derivative of bile acids, which come from the liver when you ingest a meal, and it's released into the intestine to break down the food. And then the other is a really small little molecule that is released upon ingestion of fiber in your diet. And so those two things together seem to actually together cause a greater increase in P-glycoprotein. And so those could potentially be given. No, there was work recently looking at that small molecule by delivering it directly to the intestine via enema. And there were some conflicting results on the impact to patients, but it's potentially an area for improvement.
That's interesting. Thank you.
Okay, great. I think we have no more questions and we would like to wrap up here. Thank you so much, Sage and Benedikt, for such an awesome talk today.
Well, thank you very much. Thank you for your questions.
Yeah, thank you
thank
so
you for
much,
having
everyone,
us.
joining in. And thank you, Sage and Benedikt. That was very interesting, and we learned something really new for most of us here. So yeah, that's exciting. We do want to send out our surveys, so please do fill it in and let us know if you have any suggestions or any advice for us, and whether you have any topics that you would like to hear from us about as well, so we can organize those talks for you. And we definitely want to say that our next event is going to be on October 27th. So we will send out the information soon, and we hope to see you then. Thank you very much, everyone, for joining in.